and then they induce vascular dysfunction. Directly moving to our research questions, we asked whether inflammation induces epigenetic changes. What is known between crosstalk between inflammation and then endothelial cell function, which is mediated by epigenetic changes. Mostly we are working with DNA methylation. So when we talk about inflammation, we say about IL-6 because IL-6 is known pro-inflammatory molecule which is elevated in many diseases which is associated with chronic inflammatory conditions or sterile inflammation such as cancers, diabetes, metabolic disorders, arthritis, autoimmune disorders. In many conditions, IL-6 is involved. So we thought to understand how an inflammatory molecule can induce DNA methylation changes, and particularly in endothelial cells, because I said you the reasons for endothelial cells. So we started with very basic experiment where we treated endothelial cells with different amounts of IL-6 in different time course manner, and then we understood try to do levels of methyl cytosine level in by HPLC. We found that IL-6 can induce hypomethylation state in endothelial cells, which means that there are genes which can be overexpressed in response to IL-6 via epigenetic changes. So now the next thing what we did is we did Western blots to see whether DNMT isoforms in humans can be modulated by inflammation. Yes, we can see a time course and those dependent assays both says that DNMT1 and also DNMT3B are regulated by IL-6 but not DNMT3A in a time dependent manner. So basically these get degraded. Now what is this degradation? That is due to proteasomal degradation, because when we treat the cells with MG132 and proteasomal inhibitor, it brings back the levels of DNMT1. So we say that IL-6 degrades DNMT1 by proteasomal degradation. To understand what are these genes which are involved, we tried to do a microarray assay, DMH based microarray assay. We treated the cells for 36 hours and then did with IL-6 and then did DMH assay, and then found many CPGs with their hypomethylated and all of this, which were basically related to either angiogenesis, right, or insulin signaling, which we see. And most often we try to find out which are these genes which are regulated by STAT3, because we wanted to connect IL-6, STAT3, CPG, and then gene expression. So we found out many such genes which harbor in many of these promoter elements, and tried to validate these genes in these cells, and then found RSPP6 kinases, PI3 kinases, and then F3 kinases, interleukin, growth factor 1, IGF-1R receptor. Many of these are regulated by IL-6 and then epigenetically regulated. This is what we showed. Finally, interestingly, we found VEGFR2 as one of the IL-6 determinant. IL-6 also induces VEGFR2 expression via DNA methylation. Now, when a vascular biologist finds VEGFR2, it's like going to Mecca and Medina, because these are very important molecules for vascular biology. So we hold on to these molecules, and then we try to understand whether these are important. These signatures are important for disorganized angiogenesis in human breast tumors. Then in that thought, we isolated endothelial cells from malignant and non-malignant part of human breast tumors, which are clinically characterized, and they are all ER/PR positive cells. Now we made a protocol for this, which is very much reproducible, and then we extensively characterize these cells. Now normal endothelial cells are 5 centimeters apart from the same tumors, same patients which we get. So from each patient we try to isolate 4 different cell types. One is myoepithelial cells and endothelial cells from malignant and non-malignant parts, and then we were very much successful. We have got some bank of at least 25 patients by now, and then we characterize extensively by many markers, endothelial specific markers, stem cell markers, and then non-endothelial cell markers to avoid all of these contaminations which we have. We tried also to do a karyotyping to rule out vasculogenic mimicry which happens in endothelial cells, because to say that okay these are most endothelial cells but not which is coming from a tumor cell per se. We again characterized by their functional assays like proliferation assays in response to sorafenib, which is a drug, anti-VEGFR, anti-kinase drugs, and then in both 2D and 3D cultures, we did many assays to prove that these are very pure populations of endothelial cells which are coming from breast. We also characterized tumor endothelial cells, tumor epithelial cells, myoepithelial cells by CD10, and then estimated levels of IL-6 and then VEGF-A, and then we found that they are highly expressed. These are again characterization of normal and tumor endothelial cells. For IL-6 signaling intermediates, we see and many changes in between normal and tumor, and also we see changes in DNMT expression as well, and also VEGFR2 phosphorylation and total levels of VEGFR2. Now, by now we can say that these are pure population of endothelial cells and myoepithelial cells which we have identified from these cells. Now we did paracranial interaction studies where we treated conditioned medium of myoepithelial cells of the tumors which are coming out to the patients, and then which contains IL-6 or VEGF, and then treat them on WHOX to see whether VEGFR2 upregulation is there or not. So we see very good upregulation of both constitutive levels and then IL-6 induced or conditioned medium induced levels which is there, and also with DNMT changes we see. Then we did functional assays. This is what we wanted to do, that is to make a model for disorganized angiogenesis. Now until now the angiogenesis models are against sprouts. So it means that but it doesn't happen same thing with FGF, which is another pro-angiogenic molecule. So which shows that IL-6 is very specific to make disorganized angiogenesis in breast tumors. So here we can say that IL-6 abrogating IL-6R by IL-6R antibodies can again bring back to the levels of these ones, and then they started responding better to VEGFR2 antibodies. So here we can see IL-6 can induce more leaky blood vessels. Here they are not making blood vessels, but when we make the titrations of VEGFR2 antibodies in different concentrations, now they can again come back again. These VEGFR2 antibodies can make again the blood vessels, the blocking. So kind of vessel normalization is what we did. So until now the summary is that epithelial cells which is coming from tumors, they secrete more IL-6, which acts on IL-6R, which leads to proteasomal degradation of DNMT1, which makes hypomethylation of VEGFR2, and then that gets sequestered to the membrane, and then we have disorganized angiogenesis. And then if we block, then we have again normalization of the blood vessels. Now, as we all know, most of the angiogenesis drugs are targeted towards VEGF and VEGFR2, but they are not. Most of the patients do not endure with this because only some population of patients can respond to these existing VEGF anti-VEGF therapy because of many of the reasons like resistance which they develop. So now the next question what we asked was, are there anything which are proteins which are regulated by IL-6, epigenetically controlled, but in sprouts over there? The next few minutes I would like to just talk about diabetes, although it is a cancer conference. But this is what we do related to DNA methylation again in these endothelial cells. So understanding the functions. Here we are trying to answer whether DNA methylation plays an important role in metabolic memory. So this metabolic memory is a process where despite of glucose normalization in diabetes patients, they still have complications of vascular complications like retinopathy or neuropathies or cardiovascular diseases. This is known that this is due to most of the DNA methylation changes, which is permanent epigenetic change which happens. But unfortunately or fortunately, we do not know the reasons for all of this. So that's why we took this project. These are all clinical evidences to show that there is an evidence for metabolic memory. So many of the extrinsic factors do DNA methylation changes in endothelial cells, which leads to these vasculopathies. So the first experiment what we did is treated with high glucose, and then we see that there is an upregulation of DNMT1 in glucose dependent manner, and also of both DNMT1, 3A and 3B, but 1 is very potential, but 3A and 3B changes due to the time and doses. Also in response to advanced glycated end products, we see more upregulation of DNMT1. We did an assay to mimic metabolic memory. Here we treated for 24 hours, we treated with high glucose for 5.5 millimolar, different concentrations of high glucose, 15 and 20, and bring back to 5.5 for next 48 and 72 hours. So despite of glucose normalization, we still see there is more of DNMT1 which is upregulated here. And these are interestingly, these are next generation of the cells. These are split and then again grown to the next class. So transgenerationally we see more DNMT1 upregulation which we see in endothelial cells in response to transient glucose. Which is similarly we also see in some of the in early time points at least we see also in aged products, advanced glycated end products. So we developed mouse models by high-fat diet models, and then in iotal cells we see again despite of reversal of with the normal diet for 10 weeks, we still see more DNMT1 upregulation over there, which is there in case of high-fat diet, but also after 10 weeks after reversal, we still see that memory over there. And then in case of functional assays also, we see these phenomena. In these conditions, we did metabolomics analysis, where we have 4 conditions. One is control, and one is DNMT1 overexpression, and then high glucose, and then high glucose which is bring back to normal levels. And then we see that there is a change of huge change in nucleotide metabolism. Endothelial cells are quiescent in nature until unless we have physical and physiological and pathological conditions. They don't proliferate, but in case of high glucose they start proliferating, and then we see more of high nucleotides which are still upregulated in despite of glucose normalizations. So we in the normal endothelial cells we see this where the interspecies cycle the cell and give energy, but in case of this metabolic memory, we see the cells goes to more of proliferative state, and then more of these nucleotides gets upregulated. So in these conditions, we did an RRBS analysis to see which are the DNA methylation signatures which are there in the metabolic memory. So the whole genome bisulfate analysis, and then we see that there are many genes which are important in endothelial functions have still sustained DNA methylation levels which are there. And then these are all different pyrimidine and purine nucleotide synthesis genes which are correlated to these ones. And finally, we ended up with a protein known as IMPDH2, which is in the crossroads of salvage and de novo synthesis of DNA synthesis, and then which is sustainably, which is sustained, which shows sustained DNA methylation on its promoter, despite of normalization of glucose. So now we again come back to animal models. And then we show that the IMPDH2 and DNMT1 are still more, and then that comes back when we have other cytidine treatment. So which means IMPDH2 is regulated by DNA methylation in the iotal endothelial cells. We also looked at diabetic retinopathy models, and then we see that there is more in case of retinal tissues, also show more of IMPDH2 and then DNMT1, and then we finally repeated patients to show that diabetic individuals with proliferative retinopathy has more IMPDH2 levels. This is in blood, and also this is in vitreous exosomes, which is upregulated in the tissue specific manner. And then when we add up the IMPDH2 inhibitor, mycophenolate then DNMT1 gets restored, gets down-regulated in response to these ones. So these are all our collaborators, and I thank all my students who have generated this data, and also these are funding agencies which have provided enormous support for